Okay, so now we're going to go into glycolysis and cellular respiration, and we will spend the rest of the semester talking about some of this pathway. Just to set us up, glycolysis, as you can kind of tell for the name, the first part, glyco, stands for glucose, and then lysis means to split. So the purpose here is to split a molecule of glucose and pull out some of the energy in the process. Every organism on the planet undergoes in glycolysis as a way to initiate some of this energy extraction um, um, mechanisms. And so this is a basic mechanism of how glycolysis works. Obviously, we're going to get into details. This is a 10-step process, and we'll go through each of those steps. But globally, we can simply say this. When you take a molecule of glucose, there's a lot of potential energy in that glucose, and so the whole purpose of cellular respiration is to extract that energy. We're going to invest 2 molecules of ATP, so we're putting in our own energy in order to destabilize this glucose molecule. This unstable molecule can be split down the middle, and as a result of that splitting, we will gain 4 ATP and 2 molecules of NADH, and then an organic compound, pyruvate, which is a 3-carbon structure. And so here we invest 2 molecules of ATP, we gain 4 molecules of ATP, so we have a net gain of 2 molecules of ATP, 2 molecules of NADH, and this organic compound, pyruvate, which has a lot of energy associated with it on its own that we will pull out during the last 3 steps of cellular respiration. Um, and so all we're going to do is kind of go through these processes in order to see how that happens. Glycolysis takes place in the cytoplasm and occurs in the cytoplasm, and it does not require oxygen. So here it can happen without the use of oxygen. All the other processes that we'll talk about, pyruvate oxidation, um, the citric acid cycle, oxidative phosphorylation, they will use oxygen at some point. And so they won't be initiated unless oxygen is present in the cell. And this is just a Gibbs free energy look at what's going on. We have this glucose that has X amount of energy associated with it. And then we're going to raise the Gibbs free energy by adding ATP on our own. And then we will crack it. And when we do, we're taking something that is complex, making it more simple, right? You have something that is 1, you're making it 2. So that's an exergonic reaction. You're going to release energy, and then we're able to capture a fraction of that energy in the form of NADH and ATP. Um, this is obviously not all the energy that is made from the oxidation of glucose, but we are able to get a fraction of it that we can use, that the cell can use on its own. And so this is a look of each of those 10 steps um, from our book. We'll go through all of these. I'll kind of break it up into the prep phase and the payoff phase. I'll give a broad overview now, and then I'll pull out some of the enzymatic mechanisms that I think are interesting and that go back to our first principles of enzymes, of how these enzymes work. If we were taking Dr. Young's biophysics class, he would spend the rest of his time really delving into the mechanisms of these enzymatic actions. I will say, for our purposes, recognize that these are all enzymatic actions, and remember what an enzyme does. It speeds up the rate of the reaction, but it doesn't change the delta G. So the delta G of these particular reactions, or the way in which that reaction would occur, is unchanged. All the enzymes do is speed up the rate. Another kind of key thing to point out is that after we invest a molecule of ATP in the third step of glycolysis, we do not add any more energy into the system at all. We don't invest anymore. Um, Everything else occurs from the downward flow of breaking down this glucose molecule and pulling energy out of that glucose. Now, there are certainly steps that are endergonic, right? And typically, when we have endergonic reactions, we couple it with an exergonic reaction. But in order to couple it with an exergonic reaction, you'd have to add energy. You'd have to provide energy some kind of way. And so we will see the cell has tricks to avoid adding more energy into the system after this third step. From here on out, everything that we do, all the way to oxidative phosphorylation and creation of ATP through ATP synthase, we don't invest any more ATP or any more energy into this system. So that's an important thing to note. And one of my favorite test questions is, how do we get past this energonic step if we aren't adding any more energy? And 50% of the class will say, well, you couple it with an exergonic reaction. And that gets obviously a big red X because you can't do that if you're not spending any energy in the process. Okay, so let's kind of go through the steps of the glycolysis, and I will say it wouldn't be a good biochemistry class if you don't have to know the products and reactants, so the intermediates of this metabolic pathway, the structure of these compounds, and the enzymes that catalyze the reaction, right? And again, I think I said this before, but there are a couple of ways to do this. You can brute force remember 10 different compounds or 10 different intermediates, or you can recognize the process and simply know the chemistry of what's going on in the next step so that you remember one structure, glucose, and then you know that the first step in glucose is going to be the phosphorylation in carbon number 6. So you just put a phosphate group on that glucose. And the next step is isomerization, so you just change the ring structure, right? And so by understanding the logic, then you kind of get away from brute force memorization, and then you can never be led astray in terms of why each step is occurring. The last thing to point out for glycolysis is that this goes by the principle of Occam's razor, right? So that the simplest pathway is the one that nature will go with. And so every step has a purpose. And there will be steps that don't seem to make sense, but they pay off 2 or 3 steps later, right? And so there's a logic and purpose to every step in the glycolytic pathway and later on in the citric acid cycle as well. All right, so let's do kind of broad strokes overview as to what's going on here. Obviously, first we start with glucose. The first step is to take a molecule of ATP and then use a phosphate from that ATP and add it to glucose. This happens at carbon number 6. So you form from glucose, glucose 6 phosphate. Now, this will happen whenever glucose comes into a cell. This is an investment in energy, but there are other pathways for this glucose 6 phosphate outside of glycolysis, right? So there's a lot of good reasons as to why the cell will invest this energy. Um, it makes it easier to keep the glucose in the cell. So the phosphate group is negatively charged, so that glucose 6 phosphate would have a harder time leaving the cell. But a big one is that glucose 6 phosphate is a different molecule from glucose. And so glucose in the blood is sitting around without a phosphate in it. So by changing glucose, you decrease the concentration of glucose inside the cell and therefore increase the concentration gradient between the blood glucose outside of the cell and the glucose concentrations inside of the cell. So you're able to, by investing that 1 ATP, you're able to drive the concentration gradient in a way that you were able to pull more energy out of the downward flow of glucose into the cell from the external environment. So this happens kind of automatically. This does not commit the cell to the glycolytic pathway um, per se. All right, so we've added that one glucose to form glucose 6 phosphate. The enzyme that does that is hexokinase. And remember, enzymes are named after their activity. So the kinase activity pulls a phosphate from ATP, and then the substrate here is glucose, but another word for glucose is hexose, so hexose kinase, right? And again, if you don't know what you're doing, you can always kind of look at the enzyme name and try and figure it out. So the next step is to isomerize this glucose to form fructose 6 phosphate. So all we're doing here is changing the ring structure, right? So in isomerization, we still have 6 carbons. We aren't removing carbons, but we're changing this ring structure, and we're actually taking the aldehyde of glucose and making ketose out of it. And we'll see that in a little bit. The enzyme that does that is phosphohexose isomerase, right? So it's an isomerase, and what is it working on? A phosphorylated hexose, right? Which is glucose 6 phosphate. Okay. The next step is to take this fructose molecule and then phosphorylate it again. The enzyme that does that is phosphofructose kinase, right? So it's kinase, uses a molecule of ATP, and then it's working on a phosphorylated fructose molecule, right? Now this step again, we're using ATP, right? We're investing energy, and this step commits uh, the cell to a glycolytic pathway for this molecule. So this is a step that's going to be heavily regulated. So phosphofructose kinase, the enzyme, is one that's subject to a lot of regulatory control. And we'll see that in probably the next lecture or something like that. So uh, this is an phosphofructose kinase is an allosteric enzyme, and it can be activated by things that indicate that the cell is at work, and it is downregulated by things that indicate that the cell is at rest. And we'll see that in a little bit. The next step is to take this fructose 1,6 bisphosphate, and we're going to split it down the middle to form two 3-carbon structures. The enzyme that does that is aldolase. This is because this is a inverted aldol condensation reaction. These two 3-carbon structures are going to both have each have a phosphate associated with it. And so now we formed two 3-carbon structures. Glyceraldehyde 3-phosphate is 1, and then dihydroxyacetone phosphate is 2. Um, this is an aldehyde. Obviously, this is a ketose. The enzyme that does this is triose phosphate isomerase, right? So it's an isomerase here. Oops, no. This isomerase activity is going to convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So it's going to take a triose phosphate, dihydroxyacetone phosphate, and then convert it to glyceraldehyde 3-phosphate. Now, in doing so, it kind of cleans things up a little bit. Instead of dealing with two compounds, you're now only dealing with two molecules of glyceraldehyde 3-phosphate. And so now you can use the same enzymes and the same pathway, and you just have to double everything. And so all of that to form these two molecules of glyceraldehyde 3-phosphate is the prep phase. And then once we form these, we go to the payoff phase where we can actually extract energy. So 3 of these 5 steps are actually going to pull off energy in some form, either NADH or ATP. So here we have our 2 molecules of glyceraldehyde 3-phosphate. Next thing that's going to happen is that we're going to use 2 molecules of inorganic phosphate. So this is not from ATP, and we are going to reduce 2 molecules of NAD plus to form 2 molecules of NADH, and we're going to form 1,3-bisphosphoglycerate. And so here we have 2 phosphates associated to this molecule. The next step, oh, and the enzyme that does that is glyceraldehyde 3-phosphate dehydrogenase, right? The substrate, glyceraldehyde 3-phosphate, which is made up there, and then the activity, A dehydrogenase pulls off electrons, right? And so this is going to take electrons from that glyceraldehyde 3-phosphate and put them onto something else, in this case NAD plus. Um, then we have 1,3-bisphosphoglycerate, which has those two phosphates in it. We're going to extract one of those phosphates to form ATP from ADP. And that is the first substrate level phosphorylation event. And the result there is going to be 3-phosphoglycerate. So you have your phosphate group attached to carbon number 3, and then you've lost one of the phosphate groups over there. The next step is to uh, the enzyme that does that is phosphoglycerate kinase. Right here, we're taking the phosphate from 1,3-bisphosphoglycerate and putting it onto ATP, but enzymes don't have a directionality to them, so they can go either way. All right, 3-phosphoglycerate, we're going to isomerize this, and technically, since we're moving a functional group from carbon number 3 to carbon number 2, this is a mutation or mutase reaction. So phosphoglycerate mutase takes that phosphate, puts it on carbon number 2. This actually allows for some good chemistry to happen later on. This 2-phosphoglycerate gets a water removed from it to form phosphoenolpyruvate. And then this phosphoenolpyruvate is the phosphate is removed by the enzyme pyruvate kinase. This is another enzyme in which there is a lot of regulation behind it. The removal of that phosphate forms pyruvate, and it forms 2 molecules of ATP. And so as a result here, uh, the this process, you have minus 2 ATP, you have plus 4 ATP in steps 7 and 10. You have plus 2 NADH. And so the net is 2 plus 2 ATP. So that's energy that the cell can use directly. And then plus 2 NADH, which the cell has to process in order to use further. And that happens during the citric acid cycle. So that's glycolysis in a nutshell. I'll stop here, and then on the next lecture, we'll go through kind of detailed processes. I'll pick out some of these mechanisms to to discuss in detail. All right, see you soon.